Here we go. Okay. So this is actually the final topic we are going to cover today, which is the Hi-C. Again, I don't know whether we have already covered this in our lecture or not, but I think I noticed there are recorded version of the lectures on this topic. So feel free to check it out. So here I just post a very brief history of this technology, starting from now it's like 20 years ago, the first version of trying to get this chromosome conformation, and that at that time it's called CCC, so 3C, where you're trying to pick one gene versus another gene. You're trying to look at the pair of the genes or the regions, and to trying to see if you have any interactions between the regions. Yeah, by the way, Hi-C is just this technique, and trying to imagine in your nucleus all these chromosomes are completely a mixture. And the hypothesis here is that some of the genes can interact with each other, or some of the genetic elements can interact with each other, even across chromosomes, by this spatially getting closer to each other and trying to interact with each other. And then the basic idea behind all these techniques in Hi-C, 3C, 4C is that we artificially introduce some of the cross links, when all the chromosomes are in a mixture state. And then the chromosomes that are closer to each other would have more cross-link events happening. And then now you lysate it again, you digest it into fragments, and you do sequencing. And then the difference between the 3C, 4C, 5C is different ways to identify what the sequence are. The sequence can be array-based, or can be qPCR-based, which is 3C actually, or nowadays a more modern way, more than 10 years ago, or 10 years old already, more modern ways to do this next generation sequencing, so that you can directly take the entire sequence of the cross-linked pair of genes. And then you can imagine the reads now we have is would include two parts, one from gene A and one from gene B, and they are cross-linked with each other. So now what should we do with this kind of data if we want to reconstruct their original structure? So imagine now our input are gene A and gene B fused reads, and two, we know where this gene A is originally from, and we know where this gene B is originally from. Because we have the reference genome, we can map it back to the reference genome. These are the two information we have, and the output, the result we want to have. The question we are asking is that originally, spatially speaking, which of the genes are closer to each other? And the idea behind the hypothesis behind this is that if two genes are more frequently closer to each other in the spatial in the space, then these pairs of genes are more likely to have regulatory effect between each other. Okay, which is still an open-ended question, but that's the idea behind it. Okay. Sorry. Yeah. So for this one, we would need a package called run Hi-C. Namely, it's a package for Hi-C. So that would require multiple different dependencies as the supportive package. So I would say I would not recommend you to try this on your local machine. So try to do this in on Canon. Use this environment that we generated already for you. So like we said here. Sorry. So in the homework folder, you have multiple different environments. So for this question specifically, we want you to focus on this Python 3.7 version, because that's what the run Hi-C requires. So you do activate this in the environment like what we did before. Activate, and you copy the path here. Okay. And you copy this, and you also load. Before you are able to work with Conda, you also need to load at Anaconda at Python before you're doing this. So always remember to load this before you're doing that. I'm doing here is because I've already loaded this before. But if you do not, it tells you basically, I don't know what count that is. Okay. And also similarly, you also need some genomic bioinformatics tools to support. Some of them include SAM tools, Minimap, and BWA. So the BW here is a kind of an alternative version of the Bowtie. It's a index generation software. So we also put in the homework asking, what is the the difference between BW and Bowtie. So I wouldn't cover too much here, but just very briefly, that would really, each of them would have different scenarios that each of them would be better fitted to. I think one major factor people concluded, empirically, from experience, is the length of the race. Okay, I'll just put a hint here. We also put a few additional reading materials in the question here. Actually, I think I put it here. Yeah, so trying to read through their conversation and to get a really understanding of the history, and also get an understanding of what the question is really about. Okay, so now after you you load this environment, after you understand what is the difference between BWA and Bowtie, another optional thing we would again recommend you to try is trying to build this index, use BWA, which probably you have already done before, because in homework 3 you are using this BWA, whereas in homework 1 you are using this Bowtie. So you actually have already finished both. But here, I just want to give you to clarify what the difference between them. Okay. After we finish all this, again, this is optional. You don't have to do this. We have already generated the index for you. You can load it directly from a similar folder as and it's a HG38, but now this time under the P2 data. Okay. So this folder will look something like this. So if we go to that folder, this is the kind of the prerequisite for run HiC. Another there are quite a few artifacts of this run HiC, because this run HiC is not a it's being actively maintained, but it's developed by a single contributor or quite a few contributor. So try to be be cautious about their document, and when you're trying to use their comments and tutorials. And for example, here the reason I'm saying this here is because imagine actually we allowed HG38, the genome, to be in FA or to be in FASTA file. They are actually essentially the same, but this run HiC requires it to be FA, and FA has to be. There's no way to specify this actually. So this is just one type of artifact you would have, you might encounter when you do this homework. And that is also in part the reason we recommend you to start working on this homework earlier, because now we are looking at a lot of third-party packages. So once you get all this index file and all this reference data prepared, some of them are from the Canvas, some of them are from the shared folder. After you set up your working directory, remember to put everything in the same folder, in the same directory, which is a second artifact. So it requires you to put everything all together. There are other ways to do it, but we don't recommend. So try to put everything in one folder, so that makes your life easier. And then if you have your working directory looking something like this, you are good to proceed. If you don't, go back to the previous steps to see wherever that is off. Okay. After you prepared for this, you would be directly able to call this run HiC. Now, let's go to the document so that you know what I'm talking about. So this is the run HiC document. So it is also asking you to prepare for the data, which we have done for you to prepare for the genome index and the mapping. Now we are doing this mapping thing, using this run HiC mapping. Remember to call this run HiC only after you activate your content environment. Otherwise, you wouldn't be able to, like your system wouldn't be able to recognize this command. And now it will require you to generate some of initial files for like the metadata configuration. And you can use either of this or the other, the following ones to do this mapping, okay? After you do this mapping, that would generate a folder called pairs, and you would need these pairs to do filtering. And after this filtering, it would generate some filtered folder, and you would need this filtered folder for binning, okay? Fortunately, the Run HiC authors, they have developed this bundle up question, like pile up function, so that you can directly call the function you call it once and would generate everything for you. But if you run into any issues or any bugs, you might want to go back to the previous steps to really debug step by step to see where it's really going wrong. But hopefully, you can directly call it and successfully, and that will generate you some statistical table summaries and also some plots, and you just need to put them into our homework so that we know you succeeded in working with Run HiC. And then based on those plots and tables, you can answer the following questions, those biological questions. Yeah, again here, you can do Run HiC step by step, or you can do this using this bundle function directly. Again, remember to set up your environment, have at least these 4 information. Okay, data set, the chromosome sizes, the HiC subareas, which is your input, and your reference genome, all in the same folder. Okay. Yeah, and after you run all these bundle functions, you can call this. Well, you have to call this quality function to generate the analysis of quality from your mapping and filtering. After doing this mapping, filtering, duplicates removal, we will be able to look at the actual code, that's the actual data we get from the experiments. And now this quality is giving you the first round of analysis, saying what is the quality of your HiC result, what is the percentage of this and that. And if you look into that, looking at the all these figures, would gives you a better understanding of what the HiC is really working on and what the HiC result would look like. Okay, that's all I have for this part 2.1, and that's actually the last topic for today's lab. Are there any questions? Very nice. I like this silence. The silence means we basically go over the things carefully, and we hopefully you can follow up, follow the instructions and follow the templates of the script from the slides when you are working on your homework. Okay, so in the very end, just a reminder, on this Thursday, coming Thursday, 8 p.m. Eastern time, we'll be having this Q&A session. And this Q&A session would be on the topic that is covered today. So for the other part of HiC questions and also the hidden Markov model questions, I would recommend you to save it until later, because I believe on Thursday we will be focused on the first 2 topics. And also on next Tuesday, we will be talking about the rest part of the homework and also the rest softwares that you might need for doing this kind of chromatin analysis. Okay, so if there's no other questions, I'll stop recording here.